record first screen. We need to hide the panel as well. Control. Okay. I like to use the pointer so that I'm freely moving. Okay. One last thing. All right. So good morning, everyone. This is our fourth lecture. We are moving from sequencing topics to the next topic, which is how we obtain genomic data. This is very important, especially if you start working on the review paper or start working on programming assignments. So you need the data. Without the data, you cannot do anything, right? You might be programming the best tool ever, but once you move to the real data, you figure out there are some hidden feature that you didn't take in consideration when you start programming. So you really need to get really good data before you start so that you know what you are doing, right? You know what are the things that you are dealing with. There is something happening with HeLa cells. So who remember what we talk about with HeLa and Rita-Lax? Does anyone remember the story from Rita-Lax? Yes, please. Excellent. What was it, the hospital? Yeah, excellent. Go ahead. Johns Hopkins Hospital. They replicate the cells and then distribute it to too many research centers. So it was really hard to control who get access to those cells. And then most of the companies who's doing drug development, they got those cells. And most of the cancer research is based on those cells, right? So the lady already passed away, right? But there are the family members. And then they start having lawsuit against most of those companies. So they get some of the money, hopefully, as a return for the effort for revealing the secret, the genetic secrets of this family. And this is one of the most recent lawsuits against a very famous company, Novartis, and the other one, what was it? Two famous companies, actually. This is one of them. This is public. So I posted the link here if you're interested in reading the entire thing. They claim that they develop a lot of drugs, a lot of medication out of those cells, right? And the family didn't gain anything. So this was really the source of those development. So basically, the asking for some share with the company. So it's really interesting to see that this is still happening for the last, I think, 20, 30 years, still a topic. Every once in a while, there's a new lawsuit against some of those companies. I think this will keep moving as long as those cells are still public, everyone having access to them. So it's really hard now to control and keep it secret. NIH, which is sharing those cells online, public on their database, start having some control access to some information, start hiding those some portion of those cells or portion of this genome, basically. All right, so that was just side news because now it's a hot topic in the radio. You can hear those stories very often this month and last month as well. But yeah, I wanted just to share this with all of you. All right, so now let's jump to the lecture topic, obtaining genomic data. So we have three ways to get good quality genomic data. The first one, generating those sequencing data ourselves. And we've been already talking about this for the last two lectures. The second thing, someone already did that, post it online, you could just download and use it, right? The third direction, you could simulate rather than doing the real thing. And simulation sometimes mimicking the real behavior of those data, sometimes just like some assumptions because we don't have the technology. We don't know how those machines actually work in reality. We know how the general framework, how those Illumina, Nanopore, PacBio working, but sometimes it's not about the technology, how it works. Sometimes the machine itself introduce its own error, and those errors, it's hard to predict the model of or the distribution of those error. What kind of error we are getting? Is it always insertion, deletion, substitution, and so on? So some of them did really great work trying to build the model that for the error distribution. Some of them just put some random error. Okay, we're going to see this in detail. So let's jump into the first direction, which is generating real sequencing data. And this is just a recall, a brief recall for what we were talking about. So we have the short reads, and most of those are very recent technology, especially the ONSO machine. So hopefully by now we already remembering, and we can easily recall the differences between sequencing by synthesis and sequencing by binding. Okay? Do you have any questions about this topic? Have you checked the videos that I suggested last week on how those machines work in action? What are the actual details of this machine? Okay. For long reads, we also have some recent technology, especially, so this is something from 2016, right? The other technology, it's not new, but the machine is new. This is less than 1 year old, basically. It starts getting more capability, especially in the analysis and the computation and the number of flow cells that can process in parallel. And hopefully you also remember the differences between sequencing by detection and sequencing by circular consensus. Do you have any questions about long reads? All right. I'll take that as a perfect size, the positive side of it. So we already discussed the 3 major sequencing companies and the technologies as well, which is Oxford Nanopore, Pacific Biosciences, PacBio, and Illumina, right? Those are the 3 major. However, there are other companies that they are famous. However, they are specialized in something in sequencing. They don't sequence the entire thing. One of them is 10x Genomics. You probably hear this term in conferences or on YouTube. There is a Chinese company that mimicking Illumina instrument, and there is BioNano. So if you are really curious about this, or if you choose the sequencing topic for the review paper, I would recommend you to look into those technologies as well. Please review them. Hopefully by that, we already cover everything related to generating the real sequencing data. Okay. Now let's see where we can download the sequencing data. We will start with downloading reference genome. So this is still downloading real genomic data. We have 2 directions. One, downloading reference genome. The second thing, downloading the sequencing data. What is the difference between reference genome and sequencing data? Let's score +1. What's the difference between sequencing data and reference genome? Yeah, that's true. What else? Excellent. We use it to compare those random pieces we got from the sequencing machine and rely on that reference genome to decide what is the location of each of those pieces. So we already agreed last time that those sequencing machines give you random pieces, right? And those pieces lack any information about their original location. We don't know in which chromosome, in which gene they exist, right? So we just randomly populate and generate those random pieces, right? And then we use that step called read mapping, where we need the reference genome and start comparing by every single location the reference genome and try to make a best guess. So until now, we cannot make deterministic solution, like we are 100% sure that this should be the location, but we are trying to make our best guess to say, okay, this piece probably coming from this location, and then the other piece coming from that location, and so on. Once we put all the puzzle pieces together, then we build back our reference genome. Okay, so the reference genome is something representative to our human body. So it could be, if it is for human, then to represent the human being. If it is for virus, then to represent the virus, and so on. So we could use it whenever we sequence some sample from a species or individual. The sequencing data is the thing we got from the sequencing machine. That is the basic difference between those two categories. So now let's see with the reference genome. So the best source I would recommend for everyone is NIH NCBI data sets. So that is a public data set, public database. You can access from this link. And what you get, they recently update the entire interface. So if you access it like 2 months ago, you won't see totally different picture here. The good thing with this interface that give you a lot of statistics to infer what's in there, what do they have in the database. So they have very nice interface, starting with the search. You can here filter some of the results for searching. If you are looking for very specific species, for example, you could type it over there as a name or as a simply accession number, which is an identifier for the species that you are looking for. And let's see an example. So this is some of the statistics that they have. They have about 2 million genomes, 2 million reference genomes, or let's call them genome, because reference is very specialized term. So out of those 2 million genomes, we might have a few subset that is a reference genome. So we could, for example, have 1 million genome that represent human being, right? Because we are sequencing a lot of them. We could have something from America, from Canada, from Europe, from China, and so on, right? But at the end, we will have only one as a reference genome, one that represents the majority of the population, which is something bad, actually, to have something, just single species representing the entire population, because we are different in nature. So me and my brother, yeah, very similar to each other, 99.9999% of similarity, right? But me and my cousin start having some differences, right? Because each of us inherit something from the parent, from the father and the mother, right? So as those father and mother keep changing, then the sibling or the children will have different genomic variation. So think about now, this is within the close family, right? Once we move to another country or another state or even another city, those diversities start appearing. So it's really bad that we have a single reference genome until now. That's why we start to see something called pangenomics. Pangenomics. What is pangenomics? What is pangenomics? Excellent. So we take many genomes and we build a graph out of that. Why we need a graph? Think about, I took your genome, your brother genome. There might be some differences somewhere. And then I took your cousin genome, your father genome. Every time we start seeing different mutation or different changes in those genomes. Now, if you look, I'm not sure if it is visible. You can see it? No? Every time we have this problem. How about now? Better? Yeah, it's sad for the camera. I don't think it's clear. I don't think. Maybe you can see it. But anyway, what you can see from this figure, that's the vast majority of those genomes really the same, exactly the same, right? So you can see this portion is exactly the same here as well, here, there, here, 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 and so on. Now, instead of storing those as individual data, which is going to waste a lot of resources, a lot of storage capacity, we could compress the similar portion. For example, all of this is similar. So let's compress them into single sequence. Okay? Single sequence that represents the entire population. Now we have this variation. So every time we have variation, we make a branch, make a difference, branch and keep representing those differences. So this is maybe A, this is C, this is T, and so on. So we are preserving those differences. We are not ignoring them. We are preserving them. We are representing each different path in the graph as different mutation. Those mutation may be single mutation like SNP, single nucleotide polymorphism, but could be also large structural variation, something large like 50 deleted characters, for example, or 50 inserted character. So this could still be a very large portion of data, but still we represent it as different paths. And now when we represent already those changes, we join all the branches and we continue. We have another common sequence, which is maybe this portion of data is all the same. So we represent it using single sequence. So we have the single sequence, and then we have some branches representing the differences. We join them together. We keep doing that until we represent all genomes. Now, what we have as a result? We have a single graph that is already optimized. Okay, we cannot do any optimization in terms of storage more than what we did, right? Because we compress almost everything. And we could use it as a reference genome that represents a population rather than representing a single human being. So now we preserve the accuracy of the result. We already know what exists in a population, what kind of mutation we have, right? So we have like more accurate analysis rather than relying on a single genome that represents, for example, African people only, or Chinese, or whatever, right? So for every single population, we should have that pan-genomics, or pan-genomic graph, they call it, right? So for this, you cannot see those data in this database. For pan-genomics, you should go to the Human Pan-Genome Project website. You will see those graph over there. It's public. However, they start only for human, which is the most important thing because the disease affecting us directly. I think there is a little effort with, if I'm not mistaken, with the banana tree and maybe some viruses to build pan-genome that represent those. But this still very recent topic. They started, I think, within the last 4 years or so. Okay. But whatever we talk here, we talk about linear reference genome, linear representation of those genomics. Okay. So you can, if you are interested in viruses, bacteria, any of those, you could always go to NCPI database and start accessing the reference genome. You will see here some statistics about reference genome and annotated genome. And you can see they don't add up. So those two numbers don't add up to this number. So there is a reference genome, there is annotated genome, and there is some raw data other than the reference genome and the annotated data. What is the difference between reference genome and annotated genome? Hopefully by now we agree what is a reference genome, which is the things we just discussed. Okay, what is a reference genome? What is a reference genome? You already have it on the slide. Yeah, please, go ahead. That is a pan-genome graph, a pan-genome. What is a reference genome? If I say pan-genome, then I mean the graph. If I say reference genome only, I mean the linear genome, which is single of those, any of those. If it is representing a population, we call it a reference genome. If it is not representing a population, we just call it genome sequence, human genome, and so on. However, if we use it to represent the entire population in Georgia, for example, that is a reference genome. Okay? Do you remember the thing we discussed about 2003, the consortium effort with Japan, UK, Europe, US, cost $3 billion to build that genome? That's the thing we call reference genome until now. That is still the reference genome that everyone is using. Okay? That's what we call human reference genome.
like continents? Do we have those right now?
Excellent question. So once we complete the reference genome in 2003, then each country starts having its own initiative to do sequencing for its own population. Why to do that? To understand what kind of disease we might have in the population. Why we need that information? To plan for the future. So what kind of hospital do we need to build? Do we have a lot of cancer patients? What kind of mutation do we have in the population? What kind of medication do we need to develop for our population, right? Rather than just to develop medication for random people that we don't know where they are living, it's okay to focus on the population, right? Because those mutations develop over time. And over time, there was some environment factors affecting the living or affecting the DNA. Could be food, could be the way we live, and so on. Some radiation next to us, all of those factors affecting the DNA, right? So if we develop personalized medication, it might be very suitable for people living here, but not suitable for people living somewhere else. And we already see example with OpenSNP database, right? That's an excellent question. So we have 1,000 genome project, for example, that represent population in the US. We have also started in the Middle East. It's very popular these days. Qatar, Saudi Arabia, Emirates. We start seeing a lot of grants for those projects, just to try to understand the population, because the population over there is very small. So it's easy to capture those. Here, it's a few million people, like, it's really hard to understand everything happening. But it should start some initiative with the rich states, for example, California, New York. That is easy job for them to understand the population, what's happening.
Is there a reference for every?
Currently, no. Currently, no. It's really difficult to afford the price to do that. I would imagine UK, they have it. US, they already having the biggest effort in that regard. I would say Saudi Arabia and Qatar, very ambitious in terms of the number of people to be sequenced. But whether that effort is good, publicly available, all of this we don't know. So normally they hide those secrets, so they don't want to reveal what's happening. So Turkey, for example, started something, I think 7 years ago, to start sequencing 1,000 individual and understand their genome and so on. I think they stopped the project 3 years ago or so. There is no much result in that direction. I don't know what's happening. They have a lot of money, right? They could do it. But I don't know what exactly happened. Political issue, normally, yeah, as in any other country. All right, so what is the difference in terms of annotated genome and reference genome? So reference genome basically is annotated, okay? So it's already annotated. However, the reference genome is one of those. So we choose one of the best genomes and we consider it as a representative for that population. And then once it's representative, we call it reference, okay? And we keep updating that. So once we choose it, it's not like we just have it and done. No, we keep improving it, okay? So likewise, what happened with the 2003 genome, we published it like almost 20 years ago, but we're still improving that until today. So we discussed last time the 2022 paper where we improve, and we have the most complete reference genome out of it because of hi-fi reads, for example, and some other technologies. Yes.
So you said that the best one. What is the criteria for it?
Excellent question. So best here means the most complete one, because once we sequence those reads, right? We discussed last time about a very difficult region in the chromosome to be sequenced, which is around the center over here. Normally, this is very difficult to read, right? So once we have this portion of data, we have it as a gap. So we could read those tails or the arm, we call it, of the chromosome. But once in the center, it's just gap, just empty. We don't have information here. So most of those annotated genome, they have gaps. That's one. Second thing, the quality. If we use nanopore reads, we expect to have a lot of inaccurate bases. Okay, so although we have them in the genome, but they are inaccurate. So we don't have high confidence to say, no, this is A, not C, or this is G, not T. Okay? So this one is the most complete one, the most accurate one. And we have a lot of studies on how to refine or fine-tune each of those bases until we are really with high confidence knowing that this is the right content. But again, this is for that individual that we choose to build the reference genome. So it's still representing single individual. Although in 2003, it was about, I think, 19 individual, 70% of the entire genome we choose over there coming from one individual from New York. And the other 18 individual they choose, also living in New York, but they contribute little to that genome. So still 70% of that genome coming from single individual. And we are using that reference genome for the entire world, not only for US, for the entire world. Because that time it's costing a lot, $3 billion to get only that single genome. So we cannot keep doing that for other individuals, okay? But it was really fueling a lot of effort after that. So we start understanding what is happening with the genome. Although we make a lot of assumptions, we assume that this is sharing similarity with any other human being. That's why whenever we sequence a human individual, we got the reads, we compare to that reference genome. This is assumption, right? Assuming that we will get something at the end. Okay, hopefully we understand what's the difference between annotated genome and reference genome. But what is annotated? What kind of annotation we are adding to the genome? Excellent. Gene information. For example, the gene that's responsible for the eye color. What is it? In which chromosome? What's the location? How long is this portion of gene, right? Those kind of information, we are recording it into the genome itself. So we have the real sequence of the genome, but at the end of the file, we start saying from location this to that location, this is gene number, blah, blah, blah. And if you are really interested in that gene, you could also look it up in the database, in the gene database, to know what is the function of that gene, what kind of protein is producing, and what kind of phenotype we could observe out of it. Phenotype is the thing that we observe from the outside. By just looking at your body, we could observe something like hair color, eye color, skin color, and so on. Okay? So those kind of annotation we will see on the genome. This is also annotated, but also well maintained, updated, and also selective one. We choose the best out of this group. Okay? That's why you can see here, very small subset out of all the annotated genomes. So if you are really interested in studying some of the species, I would recommend you to download one of the reference genomes. And the nice thing with the new interface, now they highlight which one is reference genome. The previous interface, you need to figure that out yourself. Okay? Maybe I have example somewhere. No. But once you search for something, it will give you a list of genomes, and then you will have a star next to it. It means a reference genome. Maybe we could do this in real. Maybe yourself. You have laptops, right? Do the exact same as I'm doing. So open the link, search for human, click search. You'll get table that has list of genomes, and you will see some emoji next to the first genome. I think it's in green color or blue color. Oh, you are typing the link? You just search for NCBI. I think that's the best. NCBI genome or something. On Google, you'll get the link directly. Maybe next time you could have a QR code. Or you could get the slides from iCollege. Is it done? Anyone? Yeah, click enter. Yeah, so you got the first one here. If you hover over the, no, go back. Just hover the mouse over here. Over here? Yeah. It says reference genome. Okay? So now this little, you see the tech mark, the green one? Yeah, just hover over it. It will say reference genome. So this is really very nice feature with the new interface to know which one is reference genome. And you see that out of thousands of genome, we only got one reference. Okay, that represents a human being, right? However, there are many other nice features with the search result. This is also with the new interface. Before that, it was really a mess to get some of the data. For example, if you really want only the reference genome, just tick that option. You go to the filter first, choose filter below the text box, then choose reference genome. That will make your life easy. So you'll get that single individual. Annotated genome and many other information. Over here, it's very nice feature to look for the type of assembly. What do we mean by assembly? So once we have the sequencing machine, give you a lot of you got a lot of those pieces, reads, right, out of the sequencing machine. Now you want to build your reference genome. You do read mapping, right? You got the reference genome, and you got those reads. You try to locate where I should stack them to build the puzzle or solve the puzzle. And once you do this step, you pick all of those, and you start merging them. Whenever it overlaps with each other, you merge them, okay? Until you got your linear sequence. This is a reference genome, but this is something represents you, okay? And that's what we call assembly. This is what we call assembly. Now, for some reason, sometimes we might have missing reads. So those reads might not be completing your entire genome. For some reason, we don't have pieces in this region. And then we will have something missing from that location, okay? Then this, we cannot call it a complete genome, because there are gaps, right? That's why we have different degree of assembly. So this is the most complete, means the entire genome, we have it already, right? And the smallest category is the contig. Contig is the smallest portion that we could have out of the read. So contig means a group of reads already merged together, okay? It's not read, it's not row sequencing data, it's still assembly, but we merge some of those reads, but still small portion of the genome. That's why we call it contig. And then we have scaffold. Scaffold means we have multiple contigs, we merge them together. But we might still have gaps. In contig, we don't have gap. In contig is the entire portion of the data. Like the first 1,000 genome is complete. We have it already. We are covered with that 1,000 genome. With this scaffold, we might have another 1,000 somewhere, and then we combine those two together with the gap. We still have a gap, but we call this entire thing as scaffold. Okay? So this is a contig, this is a contig, and those combined together, we call them scaffold. Okay? Then we have the chromosome, which is the thing we already know, that the human being has 22 chromosomes plus the 23rd for sex orientation, right? And the complete is the entire thing, all chromosomes together, the complete thing, okay? If you are interested in the year where they released that genome, so you could also control that. Those are very important to understand them, because you are going to use this data for the programming assignment, which is today. I'm going to release the assignment today. Hopefully you have a week or so, maybe less. I'll let you know the due date. But it's really important to understand how we're going to download this reference genome. Okay? Yes, please. Yeah.
Have another gap.
All of them, either one scaffold or multiple scaffold. But so that's a good question. So it depends on how many experiments they did. So as gradually they move, they update so they could start with single scaffold in the beginning, they report in the file, but maybe like 2 years later, they already more data from the same individual. Then they could read another region from the genome. They could append it to that file. And then they don't want to mess with the first scaffold they were reporting, so they are reporting now 2 scaffolds in the same file. and so on. And 3 scaffold, 4 scaffold. It depends also on the experiment. Sometimes they do whole genome sequencing. It means I want to read the entire thing. But sometimes for some experiment, they don't want to read the entire genome. They just want to read chromosome 1 because they are interested in certain disease that is impacting chromosome 1. So they could do some effort for chromosome 1. They already have the data, so they want to post it online so people don't need to repeat that experiment. Okay? And then later on, they do another experiment for chromosome 2. They got some scaffold from there. So they could join scaffold from chromosome 1 to scaffold from chromosome 2 and have it in one file. Okay? Again, remember, any portion of the genome you want to read, you are going to pay more money. Okay? You want to read the entire genome, you'll pay extra. So that's what is going to determine the factor whether I want to go read the entire thing or certain genome, certain gene, certain chromosome, and so on. All right. So now let's assume you got the reference genome for a human, which is the first one. You have it in the table, the first row you got as a result. And you can still see the annotation over here. It says it's a reference. Okay. Now how to download that file. So once you open or click on that link, you'll get a list of information, which is very important. You can see, for example, the name of the assembly. This is like identifier for that assembly. The taxon, what kind of species that we are reading. And also some more information like the date when it was released. This is the 2022 paper. Do you remember what we discussed last lecture about PacBio, hi-fi reads that enable us to have the most complete human genome after 2022 results? So that is the thing we were talking about, which is 2022 paper. All right, and how to download? You need to click on that button. But still, you'll get many more options or statistics about that human genome. This is the size. Once you hover, just hover without clicking. Just hover the cursor over this number, you'll get the entire number of bases in the human genome. Once you click on download, this is what you'll get. This list of options. Now, which one should we choose? I would recommend for beginning to choose FASTA file, FASTA format. Okay? FASTA is the most basic format you would get for human genome. And FASTA has the easiest representation for those bases. We're going to see an example of how it looks like. Okay? Just choose FASTA and then download. You'll get something about 900 meg or so. That is a compressed file. Once you decompress that file, you'll get about 3.2 gig. Why 3.2 gig? That is the size of the human genome. Every single character is represented as a byte or char character. Okay, a byte. Byte is 8 bits. If you do the math, 3.2 billion times 8 bits, you'll get 3.2 gigabyte. Okay. Here, the B, it's not byte, it's bases. The B is bases. How many nucleotide we have in that DNA? Okay, so 3.2 gigabases. Okay, which is in math 3.2 gigabyte. Once you download it, you will see this is I copied from the file itself, the header of the file, just the few first lines of the file. That's exactly what you will see once you download it. This is, I think, the first 1, 2, 3, 4, 5, 6, the first 7 lines of that file. That is the thing we were talking about. Do you remember the gaps we were discussing? This is the gap you will get. You see all of that? N. N means I cannot determine if this is A, C, G, T. So I just have N. N means undefined, unsequenced, something like that. Like we don't know what's the content. We just have N. Okay, we don't have confidence on how to read that content. But I think after 1,000 lines or so, you start seeing the content of the genome. You start having a lot of A, C, G, T. The first 1,000 line, just basically N. Okay. What we have here, the information of the first chromosome. So we are talking about chromosome 1 only in this portion of the file. But once you move next, you will see chromosome 2, then 3, 4, and so on. Here are some statistics also for the human genome. All right. So let's see the details of what we call as a FASTA format. FASTA format. You can call it FAST-A or FASTA. FASTA is the more professional way to say it. Okay. So what is FASTA format? FASTA format first is a text-based format. What does it mean? When we say text, it's something human readable. Okay. It's not encrypted. It's not encoded. It's just text-based. What does it mean? The things we saw. You can read it with your naked eye. You don't need to convert anything, right? Just N, some details. Sounds cryptic, but it has a meaning. Okay? That is a text-based format. For representing either nucleotide sequences or peptide sequences in which base pair or amino acids are represented using single letter codes. Okay? Every single thing, genome or in the DNA, is represented by a single character. Forget about those details because it could also represent RNA, DNA, protein, and so on. You could still use the exact same format. The sequence in FASTA format begins with a single line description, as you can see in the previous example. So it should start the first line with this symbol. This is a must. You have to include that greater than sign. And that's what we call description line. So just describing what is the content that we are reading in the next line, followed by lines of sequence data. It is recommended that all lines of text be shorter than 80 characters in length. So in reality, we did this artificial division. So we divide those lines over multiple lines ourselves. We did that. However, all of this just concatenated. This is the entire sequence as is. Okay? So we divided over lines to make it easy to read the file. Rather than just reading a single line that has 3 billion characters, which is really difficult to read in any programming language, we divided over lines so we can easily navigate to which line we want to start reading. Or should we start reading from line 1, line 2, line 3, line 4? Okay? And you'll start seeing that with the programming assignment. Once you get the FASTA file, you will see it's really easy to navigate between chromosomes, jump from chromosome 1 to chromosome 2 to 3 without paying any computational cost. Okay. Now, who proposed the FASTA format? This is a professor currently in Virginia University. And when he proposed the algorithm, he was also with Virginia University. Okay? It was proposed in 1988. It's proposed to extend the format or the tool that was proposed in that time called FAST-P, which is a tool to analyze protein sequences. So he said, okay, now we have protein, we have also RNA and DNA, but we have only a format or data format for protein. So let's have something to represent all of those data. So he called it FAST-all, and then in short, he called it FASTA. Okay? This is the paper where it was, yes, please, go ahead. It depends. Sometimes it's not shown in the file at all. So it's not represented. Just this is a scaffold, this is a scaffold, and that's it. Sometimes we have N, especially if we know what is before, what is after that gap. So we have it as whole sequence, so we can represent it as N. And sometimes it depends on the base calling from the sequencing machine. We don't have any confidence. Once we capture this intensity out of the chemical reaction, like it's a blue color or green color, we don't know. We cannot capture it. It's just showing a noise in the sequencing machine or in the picture that we captured. So in those cases, what we should report? So the intensity is not going above a threshold. So we cannot say for sure that this is a chemical reaction happening, so that's why it's A, not C. We cannot say for sure. So in this case, we just report it as N. Okay? And I'm going to show you the FASTQ file. That is going to solve this problem. In FASTQ file, we add little more information to represent those cases, to tell that, yeah, this N is representing very low quality data, okay? This is the paper, if you're interested in knowing about FASTA. So FASTA is a tool, is a computational tool, plus a data format. So sometimes confusing when we say FASTA. Is it the tool itself? Is it the data format? Is it the file format? But both of them exist. All right. So this is an example, another example of the FASTA format. We have one line that we call it a header or description. And you can recognize that line by greater than sign. It's really important for the programming assignment. Please pay attention. Once you want to read that file, you need to look for those lines first. You capture them, and this will be the detail of what is next. So when we talk about this is chromosome 1, it means everything next until we got another greater than sign. That is the thing we are talking about. If we got another greater than sign over here, it means everything here between those two lines is the thing that we are talking about in that line. Okay? So if I ask you to extract chromosome 2 from the file, you should look for all those lines that start with a greater than sign, and then look inside that each of those lines and look for chromosome 2. Once you get it, you need to give me everything after that line. All right? Deal? Very easy, straightforward. All right. So that is the header. Again, we can identify it by the greater than sign. That is a must. You should have the greater than sign. And that's what we call genomic sequence. Another question, if I ask you to count how many A's we have in the genomic sequence in the chromosome 2, what we should do? We should first look for those header lines with a greater than sign, look for chromosome 2 in those lines. Once we find it, then we count within those lines, not with that line. If I ask you to count something in genomic sequence, you should ignore the header. The header is not genomic sequence. It's just information, just description. This is the genomic sequence. Again, this is combined as one sequence, but we break it down into multiple lines to make it easy to read. So all of this is the same genomic sequence. If I ask you to count how many A's in it, you should consider all of them until you figure out greater than sign or the end of the file, basically. Okay? Very easy, straightforward. You just need to understand the interpretation of those lines. Any question about FASTA format? As I told you, this is the most basic format you could use for genomic data. Just the read itself, done. There are some other older variation of that file. You will see it in practice once you start working on bioinformatics topic. However, it's very similar. You can see there are two portions of data. The first portion, some description. However, it's divided over multiple lines. And the end of this portion is the two dots. Once you see the two dots, it means the first portion of the information is finished. Okay? And the second portion of the data is the genomic sequence. However, it's organized over 10 characters, a group of 10 characters. So every 10 characters, we have a space or tab sign to divide those 10 characters together. Plus some indexing. This is character 1. This is character 51. This is character 101. It means everything before that character, 100 character in total. Okay? And so on. So you can see in the old days, they don't have too many main memory. So they don't want to repeat the computation. And they want to make it easy to read because the file in that time was very small. So in the old days, they were not able to read the entire genome. So they read small portion of the data. That's why it's easy to draw those figures because they check by eye. They don't need to build algorithm to read those files. They just open it. Okay, location number 10,000. It's easy to go to this index, check for 10,000, and we start from here. We got the piece of data without any algorithm, without any computer program. Okay? But once we start developing 3 billion character file, even more with the plants, for example, the tree, the Japanese flower, how large the genome was? Who remember the first lecture when we talk about plant? At least 50x longer than our DNA, right? It's about like 150 billion character. Those things are not useful, right? We don't need them. You cannot track the lines by your eye and try to figure out what is the location of 10,000 gene, right? So that's why those formats are already old and it's not in use, okay? There was a very famous database called GenBank or GeneBank, okay, that was using this kind of format. But now everyone tried to move to FASTA format, okay? You will see also file format. So we talk about FASTA. The FASTA, you will see it in the extension of the file itself. So once you download the human genome, you will see at the end .FASTA. So it could be uppercase, could be lowercase, doesn't matter, okay? But you will see FASTA at the end, okay? But sometimes you will see instead of FASTA, those alternatives. And it's exactly the same thing, exactly the same format, okay? The only difference is that sometimes if you use FNA, it means the file has only DNA, okay? There is no protein, no RNA, and so on, okay? If you use FNA, it means just DNA. If you use any other alternative, it has different meaning. Sometimes amino acid or protein, sometimes RNA, sometimes different kind of annotation added to the FASTA file. Okay, but the exact same thing. Right, hopefully by that, we already cover FASTA format. Now let's move to sequencing data. So we still with the second direction of downloading those data. This is very important for the programming assignment, okay? Now, as with downloading the reference genome, we need a database where we can look for some of those sequencing data. So one of the most famous databases that you could use called SRA, it's also from NIH, National Institutes of Health, called SRA, okay? So the interface you could see here is very old. It's not updated as in the reference genome. This has been for the last 10 years or so, the exact same thing. We have also here the search text box that you could use. You just need to type whatever you are looking for example, human, and then you could get something out of it. As there is always fight between US and UK in terms of genomic data, who's having the largest amount of genomic data, they also have alternative in the UK called ENA. So ENA and SRA are two alternatives for sequencing data. Sometimes you will see something unique in here. Sometimes you will see something unique in here. And sometimes you will get the exact same sample in both of them. Because those publicly available, and anyone working in a research center could upload their own data. So it's up to us if you want to upload to this and this together, or one of them, okay? That's why sometimes you'll see some common data set. Right? And we also have search result here, or a text box that you could include and try to look for something. Both of them for free, so you could access it and download something out. And as you know, currently, we have the bird flu virus circulating in the US, affecting the chicken, the egg, the milk, right? That's why we have a crisis now, increasing the prices. And you can hear it very often in the radio, right? This is how it's spreading in the US. This is from CDC website. You can access it using this. This is an interactive map, so you can click in certain states, certain particular zip code, and so on, look for infection around you, and so on. This is how it looks in Georgia State. It's really not that bad compared to California, for example, especially in the winter. Now, the virus spreading is decreasing with the winter, right? That's a good question. I don't know the criteria, but I think it won't make sense if they are going to highlight in the map if they have at least one case, because that's going to has maybe. I don't think they are doing it currently. They have single color in the map, but they have the numbers. Once you hover with the cursor over a certain place, they will show you some information. Likewise, if you click on one of the states, it will show some information. But remember, if they want to show those small number of cases, then they're going to affect the public health. So people start worrying, right? The prices start increasing because they start, okay, we don't want to, or they're going to kill a lot of those chicken, right? And then the price increase dramatically. People start consuming more milk or reduce the consumption, basically. Either they are scared from the virus or they want to get more of the healthy milk currently because in the future, most of the milk will be affected, right? So it depends. So that's why I don't think they are reporting those small number of cases. However, what I want to say here, if you are really interested in knowing that virus, the genome of that virus, you could use those databases to access the bird flu. Do you remember the example last time we discussed? If you are a farmer, you have a farm, right? And you want to study what kind of disease is impacting your plants. So you could do swap, take it to sequencing machine, get sequencing data, and then compare it to a reference genome, right? So you would do the same thing for the bird flu. So you would get a sample from infected milk or egg and so on, and you would compare it to one of the reference genomes. However, if you don't have the sequencing machine to do the sequencing, you could still get those data for free. So those are reads, not the reference genome. Remember, now we are talking about reads, sequencing data. The previous slide was about the reference genome. So if you want to get reference genome, you go to NCBI genome database. If you want to go to sequencing data, which is the read, the portion or subsequences of your genome, you would go to SRA or ENA. So this is SRA from NIH. You could type bird flu. Remember, we are looking for SRA, okay? And then you get a lot of those samples. You will see very nice filtering over here. Do you want DNA or RNA? Do you want it as a genome? Do you want what kind of sequencing machine to be used? Is it Illumina? Here, we only have Illumina because this is a very recent virus. And the cheapest way to do sequencing is Illumina. That's why you'll see only Illumina here. But as time progressing, you'll start seeing some other platform. You want a different type of sequencing, and this is the file format, which is FASTQ, because we are looking in SRA, and in SRA, we expect only to have sequencing reads. And that is the format to represent those data. If we look into other, like COVID-19, for example, okay, COVID-19 is already mature. It's been how many years? Maybe 4 years since the pandemic, right? So we start seeing a lot of those sequencing technologies for the COVID-19. So in the bird flu, we have only Illumina. Now we start seeing Illumina, Oxford Nanopore, PacBio, a single molecule real time. Do you remember SMRT? Okay, which is hi-fi reads, or the other type, which is not circular consensus. We didn't discuss it in the lecture, but this is representing two types of PacBio. One of them hi-fi, which is the high fidelity reads, or the circular consensus with high accuracy, and another type called continuous long read, CLR. Don't worry about the other type because it's not accurate as in hi-fi, so that's why we discuss only hi-fi reads. Right? So feel free to type here, not necessarily the scientific term for COVID-19, which is SARS-CoV-2. You don't need to know that in advance. You just type whatever you want over there because there is a description inside of these files. We're going to see it. As you can see from this line, you can read these things that you can understand. Likewise, with the bird flu, it's called H5N1. Okay, you don't need to remember this term. You just type bird flu at the results. Okay. All right. So now assume I'm interested in one of those. I choose based on whatever criteria you choose over here. Then I choose one of them. Let's say the first one. What you will get is this window. You'll start seeing ID number, which is identifier for SRA data. And then you'll start seeing a lot of information. The most important one is what kind of sequencing technology we use. Why this is important? Because it tells us about the characteristics of the data that we are going to get. What kind of characteristics we will get out of Illumina? The most important one, once we open the file, we will see all lines are of the exact same length. Because from Illumina, we got all reads exactly the same length. All of them. If it is 100, all of them 100. In Nanopore, we get something between 20 all the way to 4 million bases in one file. You open it, you'll see a line that is really huge, very long, right? And some lines very short, okay? In PacBio, we got something between 20,000 to 30,000. Or sometimes this is 1,000, but we filter out most of them and we keep between 20 and 30. Okay, that's why it's really important to know what kind of sequencing technology we use for this data. Plus the sequencing machine. This is important for what reason? The error model. Each of those instruments or machines produces its own error model. Sometimes the error of type deletion only. Sometimes the error is type of insertion only. Sometimes the number of those variations is different from machine to machine. If you want to download that sample or the read set, you would go to that table and you click on this. Okay? Very straightforward. You go to that table, download this read set, and you will get something in format of FASTQ. FASTQ. Remember, in FASTA is the reference genome. FASTA, it means fast all. Here, FASTQ. Okay? It means fast the query. Because this, we use it as a query. We are searching in the reference genome using those pieces. So we consider this as a query, and we try to look into the reference genome. Where does this piece exist? Okay? So we use it as a query to look in the reference genome and try to determine the location of each of those pieces. Okay? This was introduced somewhere around 2000. We don't know until now when it was exactly produced because it was never documented officially. We don't have any research paper talking about FASTQ in that old days. However, the institute was doing sequencing in that time. Remember, this is 2000. This is before the reference genome. The reference genome, the first copy was released in 2003, okay? Or somewhere around 2001 was a draft and then published in 2003. But before that time, there was very little effort on sequencing around the world. Nobody knows around this area because we don't have a reference genome to compare sequence data with the reference genome, okay? So that's why there was no interest in documenting what kind of format we should use to save this data because it was only very few places that doing that sequencing. But according to this paper, they mentioned that this guy was responsible for this kind of formatting, okay? He was the first one to introduce this FASTQ format. And you can trust it because they said, we were talking about with people from this institute to figure out who was introducing that format. So you can trust this kind of internal communication. Okay, so now let's see what are the differences between FASTA and FASTQ format. In the FASTQ format, we will get portion of or blocks of 4 lines. Blocks of 4 lines. So you can look file, you will see a lot of lines, but you can divide them over into 4 or sometimes 5 lines. But let's consider them now 4 lines, okay? What are those 4 lines? As in FASTA format, the first line is the header, where we will have more information about what is this about, okay? However, this line starts with @ sign, not greater than, but @ sign, which is the one we use in email, electronic mail, okay? @ sign. That means this is the header line. The second line is the genomic read itself, the read that we get from the sequencing machine. You can see ACGT and a bunch of N's, right? The third line is just separator, just plus sign. It separates the second line with the last line. That's it. There's nothing interesting here. Why we have that symbol over there? Because sometimes this line is very long. Imagine we have 4 million characters. It's really hard to see by naked eye what is the end of that line. That's why we just have additional line with a plus sign, some random sign. Okay? And then the fourth line, which is the most important thing in answering Grigori question about how do we know whether those M's are gap or something else. Okay? What is this line about? This is base sequencing quality. Okay? What does it mean? It means while I'm doing the sequencing, regardless if it is Illumina, Nanopore, or Hi-Fi, all of them will have that line. And that line represents the quality of the prediction. When I predicted that this is A, not C, what was my confidence that time? Is it 70%, 80%, 90%? If I'm really sure and I see high intensity in the picture coming from Illumina technology when I do the CMOS camera and capture those chemical reaction, if the intensity was high, it means I'm for sure very clear about this decision. I made it while I'm sure that this is A. It cannot be T or C or G. If that is the case, then I record something for that character. That means for each of those bases, I will have a confidence level. Okay? So if this is 100 long, then this should be 100 long. If this is 1,000, this is 1,000. And each of those bases has its own quality. And E means something over here. The F means another thing, and so on. So those are basically the quality level that I have while I'm doing the sequencing. So who's generating that line for me? Is the sequencing machine. Okay? While producing those pieces, it directly gives me what kind of level I have. So how we calculate it? How we calculate that line? So those are different characters, each of them representing something. And that representation is in ASCII code. How many of you know the American Standard Coding, which is called ASCII? ASCII code. What is it? Any definition? It's just standardizing the way that we enumerate any character. Whatever possible character in life we have, like in English, what could be? The alphabets, the numbers, special characters, right? That's it, right? What else we could have as character? Then we will have the translation of those alphabets, right? We could have in Chinese, in other languages, and so on. But in American way, they just agree on something on how they should order those characters. Should they have the numbers first, or the alphabet first, or the special character first, and so on, and what they are. Okay, so they just have a table saying that the first one in this list is, for example, A, and then B, and then C, and then D, then E, and then the numbers, and then the special character, and so on. Okay, this is just an example, and this is the actual order of it. So this is portion of the ASCII table, because before this exclamation mark, before this mark, we have a space. So space also included in the table because it's considered as a character, right? Once you type, you count it as a character. So before this mark, there's a space and there is some other character. But for sequencing data, they said it doesn't make sense to have just space in that line, because remember, when they introduced those files, they were not using computer to look into those files. They look by naked eye. And they want to make it easier for people who's looking to figure out that there is a character, not a space, or something wrong with the line. So they ignore the space and anything before it, and they start from that mark, okay? And they consider this as 0. Then 1, 2, 3, 4, 5, 10 mean the plus, okay? Then 20, 30, 40, and so on. So that is a scale representing the quality of the sequencing data, okay? Now this number, we call it quality score, okay? What does mean with the quality score? So in order to understand the meaning of 0, whether it's bad or good, we need to look at this equation. This equation means Q is this number, right? If it is 10, so you replace the Q with 10, then equal minus 10 times logarithmic to the base 10 of P. And P is the probability that the base call is error, okay? The probability that the base call is an erroneous one, okay? What does it mean? Again, so imagine we have a 10 of quality score, okay? Quality score of 10. Is it bad or good? We don't know yet. Assume this is corresponding to a probability of 1 in 10. It means in 10 times of sequencing that character, we will make 1 error. In 10 times reading that character, we will make 1 error, okay? So in 10 reads at the same location, assuming all those 10 reads exactly the same, then we'll make one of them as error, okay? And that is about 90% of accuracy, okay? So the 1 in 10 is the probability. It means 0.1 we will have here. And the corresponding quality score for it is 10. And what we are going to record in the file is the plus sign. Not the 0.1, not the 90%. It is the plus sign, okay? So if I give you a plus sign in the FASTQ file and ask you what does it mean, is it good or bad? You directly do the conversion here, and you tell me this is 90% of confidence that this is A or C or G or T, okay? As we reduce the probability, like 1 in million means 99.9999% I'm confident that this is correct, okay? So 60, as we move, as you can see, this is in green color. Green means the best quality that we will get. Red is very bad. Normally we ignore those reads if they have on average. So for each character we said we have a character representing the quality, right? In each character in that read. Once you take the average of all those qualities, like if this is 10, this is 20, 30, 40, and then 50, 50, 50, all of them, you just take the average. And if there's between this range, you just ignore the entire read. We don't want it. And we say, instead of the quality score, we say the average quality score for that read is 10 or 20. Then just ignore the entire thing. Yes? Excellent. So I agree with you. Excellent. So there are two things. The first thing is marketing. Okay, I'm able to improve the quality of my machine by this much. Now, no, your machine is 99.999. My machine is much higher accuracy. This is still significant. That's one aspect of it. I would not call it significant, especially if we talk about Illumina sequencing technology, which is 100 long reads. So it doesn't matter really, like in 1 million or 1 in 10,000, right? And then it's still accurate. But once you move from one machine to another machine, like in iPhone 14 to iPhone 15, you still have some variation to claim that this is a newer machine. Okay? So you need to claim something that's different from previous one. The second thing, it's really significant in long read. In long read, you start to have like million long sequence, right? If there is a higher chance, like between this and this, it means how many errors? 10 errors I have in that read, right? And 10 errors really significant between this and this. If we are talking about million long sequence, 10 errors really significant, especially if we are studying single nucleotide polymorphism. Imagine I'm studying a certain base and the sequencing machine giving me a different one just by sequencing error. It's not because genetic variation. So that is important. It really depends on how we look at this problem. All right? That's why, yeah, give me a second. That's why we are looking about the range. We don't care if it's 30 or 40. As long as it's between that range, it's still fine. So moving from 30 onward, it's perfectly fine. Okay? Yes, please. Excellent. Sometimes, some of the data, also you will have the lowercase in here. Sometimes you have the genomic sequence itself in lowercase. This doesn't matter. Also, if you have it in here, lowercase, doesn't matter, because the range we choose out of the ASCII table is only that one. That's the range we choose. So we don't care about lowercase, although lowercase exists in the ASCII table. So we have both uppercase and lowercase as different character, but it doesn't matter. So you just convert them all to uppercase or keep them as lowercase. Likewise with the genomic data. Some of the bad files, once you open them, you'll see here lower cases. Sometimes mix of both, lower and upper, still same thing. Okay. Yeah, definitely. You convert, please, all of them to uppercase. Okay, that's the correct way of how you represent, not the lowercase. But you know, also with this thing, this question is really a bit tricky because currently we don't have single FASTQ format. We have three. We have the original one, which is coming from the institute, Sanger Institute in the UK, right? This is the most original one. And then Illumina introduced its own formatting, especially for the base equality over here. They have different meaning for the equation, okay? Depends on their machine and how they interpret accurate versus not accurate data, okay? And we have another format. I forgot what was the company or the group introducing that. But we have three formats. But whenever we talk about FASTQ, we always refer to the original one, which is Sanger FASTQ, we call it. Sanger FASTQ, which is 2000. All right. Do you have any question about FASTQ? Okay. So what is this? This is a tool called FastQC. FastQC. What does this tool do? This is a tool to control the quality of those files. You are given a FASTQ file. You are asked to filter the low quality data. What is the first step you would do? Either you write your own program to look at the basic quality of this read, calculate the average for the entire line. So you just take the average and see whether it's 10, 20, 30, 40, the average for the entire read. And if it is below that threshold we saw in the scale, we just eliminate the entire 4 lines, not only the read. Remember, each read is represented by 4 lines. And I told you sometimes 4, sometimes 5, because some bad programmer, when they save the file, they insert a new line between every 4 lines. So you have to take care of that once you do the programming assignment. You are given a FASTQ file. There could be an extra line between every 4 lines, or might not be there. Okay. So if there is no extra line, that's a perfect file because you are not going to waste an extra line, because extra line is still a character, right? You know that. An extra line is represented as a character in the file. Then it takes space in the storage. And think about when we talk about 1 million read, that is 4 million line plus 1 extra line for each of those 4 lines. That is about 5 million lines instead of 4 million. So it's really significant to worry about that extra line. Plus, when you interpret those lines, so you have to worry about if there is an extra line or not. Because if I ask you to read every second line, means read only the read sequence, you don't want just to count 1, 2, and then take the 2, and then 3, 4, 5, 6, and then take the 6. You don't want to do that. You want to check what's in that line. Is it an extra line? Is it an empty line or not? If it is empty, you reduce your counter by 1 to take account for that extra line. So if I ask you to extract only the genomic sequences from the FASTQ file, oh, now it's erasing. I don't know what's happening. Okay. So that is the FASTQ file. We have the header. We have the genomic sequence. So this is important, the second one. We have the plus sign. We have the base quality. Okay. Likewise, with the next read, we have the header. We have the read, which is the most important one. We have the plus sign, and we have the base quality. I ask you to consider only the second line, so that is second. I think I have everything with me. That's the second, this plus, this base quality, this header. That's what we are looking for, right? So if I ask you only genomic sequences, I would take, this is the header also. This is, we can add the @ sign and the @ sign, right? I would take the second, remove the third, fourth, and if there's an extra line here, I also ignore, right? And then ignore the header, and then take that as genomic sequence and move on, okay? That's a FASTQ format. So you have to be careful about whether you have an extra line here, an extra line there, and so on, okay? Now, what this is about, this is doing this step for you. FastQC is doing that for you, plus doing trimming. Trimming mean I want to clip the read itself. I don't want the last 10 character, for example, in the read. So it can did that for you, okay? But also one important thing is to filter the data based on the quality. And this is what we see in this figure. This is the location of the character in one read. Okay? In one read, this is character 1, 2, 3, 4, 5, 6, 7, 8. Okay? And this is the quality of that character. So character 1, low quality, or somewhere between mid and high, right? Quality of 2 is high, 3 is high, 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 high. Once we arrive at the end of the read, the sequencing quality start dropping. And we discussed why in the Illumina sequencing technology, if you remember. If the read looks something like that, that is a very bad read. Let's ignore it at all, because most of the quality of the bases with very low confidence. So the entire read is useless, does not represent anything. It could be just random noise coming from the machine, okay? Because sometimes when we wash the surface, after we add the molecule ourselves, when we wash the surface, some problem might happen. Some of those molecules still exist after we wash. So they make chemical reaction on their own. That is another tool called NanoPlot. It's dedicated for long reads. Everything we talk about, short reads, Illumina example, right? So it was easy to see the bases individually, 1, 2, 3, 4, 5, up to 150. That's fine to see that. Imagine now we have 1 million or 4 million long sequences. It's really hard to track which base is causing the issue, because you will see them in dense representation. It's very close to each other, right? So that's why they, in this tool, they represent the quality in different scale. They have a 2D. So this is the read length, right? And this is the average read quality. So it's not about single base. Now they take the average of all those bases together by single number and the length of that read. So here, it's not a single read. It's all reads in the file in one plot, not single read. Over there, it was for single read. For single read, I got the plot. If I have 10,000 read, I have 10,000 plot. Over here, the entire file will be represented by single plot. And it's still very important. Why? Because I can see those are very short reads. From 100 to 1,000, I got those reads, okay? And then those, around 10,000 maybe? Yeah, 10,000 to 100,000. Most of those group of reads, okay? From 10,000 to 100,000. Now, why this is useful? I can see the average read quality. If I want below 20 or below 15, then it's easy to clip this. And based on the length, I'm going to clip this. And then I can see that the majority of the file, based on the color, because this is the darkest, means the most or the largest number of reads I'm having around this area. It means if I filter out those, I'm still good because I have most of the reads in this area. Okay? So I can use this figure to decide on which metric I'm going to filter for both the average read quality and the read length. So I can make a line over here, line over there, filter out those because I consider them as noisy read, and then use the rest in my experiment. Okay. If I don't use this visualization, it's hard for me to imagine what kind of read I will still have. Okay. Imagine I'm choosing something very significant as 20, and I'm choosing the read length as 1,000. Then I will stay only with those number of reads, right? So I might ending up having a low number of reads in my file. And then I was wondering why only I paid $10,000 for sequencing, but I got like 100 reads only. Okay. Likewise, the same tool also provides a lot of statistics, a lot of plots. It's not only about those two plots. So here, for example, you can see the number of reads that are within this range of length. So you can see the distribution of how many reads I'm getting as 100,000, how many reads I'm getting as 1,000, and in between as well. It's not like discrete. Right, by that, we're already done with FASTA format and FASTQ. Hopefully, we covered everything related to those two formats, okay? Now the time, if I ask you to convert between these two formats, what you would do? I give you FASTQ and I ask you to convert it to FASTA format. Although the purpose is different. The purpose of FASTQ is sequencing data. The purpose of FASTA is reference genome. And those two things are different. So there's a tool online. You could use it to convert between any other formats, regardless if it's FASTA, FASTQ, or anything else. Okay? So you could use something like that, or you could do it yourself easily, because you know that the first line in FASTA starts with the greater than sign, but in FASTQ with the @ sign. And in FASTA, we only have 2 lines per sequence, but in FASTQ, we have 4 lines per sequence, sometimes 5 lines. Okay? So yeah, that is something to mention. And the last thing, maybe we'll exceed the time by 5 minutes. So let's see. The last thing is simulating the data itself. If I don't want to download from any server, if I don't want to do the sequencing myself. So as you saw in FastQC, the plot of the base quality is dropping toward the end, right? So this kind of error model for any Illumina instrument, I can consider that as a generalization of the error model of this machine. Whenever I get a read from Illumina machine, I always expect to have the accuracy toward the end as very bad. Okay? And that's why some researchers build the error model for Illumina machine, and they said whenever we want to simulate a read coming from this machine, we will mimic the error in this way. So whenever in the beginning we have very high inaccuracy, okay, and then toward the middle we start having high accuracy, and toward the end we will also have again the inaccurate behavior. And that's exactly what we expect from real data. As in here, the accuracy starts increasing and then saturate, and then toward the end start dropping again. Okay? So you could build this kind of simulation yourself, right? You could impose some control until the read and make some random choices, whether it's A, C, G, T. But in fact, they don't do that. They don't do random choices. You have to provide the reference genome for the simulator. And based on this simulator, they go to location 1. They took the first 100 character as it, okay, as is. So the content of the genomic sequence is still coming from the reference genome. But what they do is that they impose the error on that sequence they got from the reference genome. So they pick the first 100 character from the reference genome, and then they said toward the last 10 characters or so, we are going to change those content, convert the C into G, T into A, and so on. Okay? So that's one way of doing the simulation. There is a very nice paper called Performance Evaluation of 6 Popular Short Read Simulators. So during my PhD time and during now, I'm using 2 of those 6 popular, and they never face any problem with them. So I would recommend them also for you if you are interested in simulation. This is the 6 simulators: ART, DWGSIM, ISS. Mason, Neat, and WGSem. The two I'm using is normally Mason and Art. I never face any problem with them. They are really quick. I think with Art, there's no multi-threading. That's the only problem. But with Mason, there is multi-threading, so you could run it very fast. You could use 10 threads in your CPU and run or do the simulation very quickly. Other than that, I would say there are some differences, especially with the error model. For example, can you specify, can you customize the indel rate? Indel is a short for insertion and deletion. So the error over there we were talking about, those normally substitution, okay? But sometimes, out for curiosity, they want to impose insertion or deletion. They want to insert some character somewhere. Can you do that with this simulator? So some of them, yes, some of them, no. For example, this, yes, yes. Here, no. Yes, yes, yes. So those kind of differences that you will have between them. You will also have something related to the speed. This is single thread. This is multi-threaded. So multi-threaded, only two tools provide that performance, which is Mason, the green one, and the blue ISS. And in single thread, you can see the speed is different. Why is different? Because it depends on how they get the read from the reference genome. If they do some computation, if they follow really strict model in doing the probabilities, or they just randomly get a copy from the reference genome and they make their own error or changes replacing those characters. That was for Illumina. Now, when we talk about long reads, the best I use myself is PBSEM3. PBSEM3. There's 1 and 2, and there's 3. 3 is the latest. 1 was for nanopore. 2 was for CLR and nanopore. And then PBSEM3 with hi-fi reads. That's why I recommend you to use it, because you can simulate hi-fi reads. Okay? Plus nanopore. So it works for PacBio and ONT. And if it works for ONT or hi-fi reads, you could also use it for Illumina reads, because Illumina is as accurate as hi-fi, or vice versa. Hi-fi is as accurate as Illumina. But that is not a guarantee, because the error model is different. The type of variation is different between Illumina technology and PacBio. All right. So I would like to give you, I think we will finish on time. I would like to give you an exercise. This is not graded, so it's up to you, okay? I'm already, you can see in the iCollege, in the content, table of content, you can see two files, dummy.fasta and dummy.fastq, okay? Those two files looks like this. This is the FASTA file, so I made my own. In reality, I copy it from another genome, so this is real data, but I make my own header over here and here. This is chromosome 1, chromosome 2. And you can see I made an extra line in between, okay? I make an extra line. And this is the FASTQ. Also, again, it's dummy. I make some characteristics in purpose between the reads. So you might see the reads are similar to each other, or might be just the reverse complement. The reverse complement. We're going to see that in the next, I think, 2 lectures, why I made some reverse complement copies. Okay, this is going to be very helpful for read mapping. What does mean reverse complement? Means when I have this sequence, right, I go to the last character, I make the complement of that last character. If it is C, then I made it as G. And this G, I put it in the beginning. So this is G. Now, when I have A, I made it as T. A, T again. C as G. C as G, and so on. So that's what we call reverse complement. Complement, because each character, I took the complement of that character. And reverse, because I just flip. What is in the beginning, I made it as the end. And what's at the end, and make it at the beginning. So horizontally flip the sequence. And that's what we call reverse complement. Don't worry about that, but I just want to tell you how I build that file, okay? I just make some tricks, not for this example, but later on we are going to use it. So hopefully we are going to use these two files moving forward with the next lectures, okay? But again, this is not graded. This is just exercise for you to get your hand dirty, as they are saying, with those files, okay? So what I'm asking you to do, write a small program in any programming language that reads only the genomic sequences, only the genomic sequences. Pay attention to that. Don't open the file and just search for those terms. What I want you to do is to count how many A's we have in genomic sequences, how many C, how many G, how many T. Remember, this is only in genomic sequences. If there is a header, just ignore, okay? You want to do that for C, G, and all of those. So this table for dummy FASTA and this table for dummy FASTA. Just count them and let me know. I have the solution with me, but I'm not going to show it. It's already in the slide, but I think when I convert it to PDF, it's not there because I hide the slides. So you won't have it, but I have it with me currently. And you have, there are two solutions, basically. Both of them correct. It depends on, for example, if you have 4C. If you have, for example, C, 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 and I ask you to count how many 2Cs I have. How many of this I have? Where's CC? This. I ask you to count CC. How many times do I have it in here? 3. Why not 2? Because I have here and here, right? There's one in the middle. Excellent. So that it depends on you how you look at this problem. Okay. So we have this, we have this. So both solution is correct. This is 2 is correct, 3 is correct. It depends if you want to look for overlapping, non-overlapping. Okay? So that's why I have 2 solutions for this. But good luck figuring out this. This is again not graded. This is not the programming assignment. I'm going to release the programming assignment hopefully by today. So you have about a week to submit it. Okay, maybe less than a week. We will see. Okay, and I still don't see any paper related to the review paper. I ask you to recommend 4 tools that you are going to read moving forward, but I don't see any. Yes, please. It's important to keep track of the same topic. So reply to yourself on the same thread. Don't create a new thread. You don't want to make it miss. So keep 1 thread per paper review or review paper. Okay. Reply and include the 4 papers, 3, 5, 6, 7. I recommend 4, but it's up to you to pick that number. Okay. Put them there. I'm going to reply if those are good paper or bad paper. Okay. I'm a bit worried because you didn't start yet reviewing the paper. I want you to start as early as possible to really provide high quality review paper. So I'm thinking to make next lecture online over Zoom just to discuss the review paper. How many of you agree? How many of you want it to be normal lecture? So I will have all of you in one Zoom session, and then could ask questions. We could go over the paper you choose. We could like filter those choices. I want to push you really to start. I don't want to keeping you until the end of the semester. Okay, how about that? How many of you agree? 1, 2, 3, 4, 5, 6, 7. About 16. Okay. All right, deal. I'll send you information on the Zoom link and all the stuff. Thank you so much for the lecture. Thanks for those online watching us. And I will see you next lecture. Next.